Okay, we can get started now this evening. I want to first thank everybody who braved your fears about the swine flu for showing up here. I did wash my hands this afternoon. I don't know if everybody else did. I'm Mike Kalichman. I'm co-director for the Center for Ethics in Science and Technology. We are, as always, delighted that we have this opportunity to be here at the Reuben H. Fleet Science Center. They have been extraordinarily generous hosts in helping us to have the programs here and to help set up. And we are delighted that you can join us this evening. A couple of things that occurred to me I'd like to know before we start, just for us to be keeping track of where we are. How many of you have not been to one of our Exploring Ethics programs before? So a fair number of people. The others are repeats, which show those of you who are new that people do want to come back again. And the second question is, how many people are actually in the field of stem cell science, which is our topic tonight? So a couple of people raising their hands. They've identified themselves now accidentally, so now we'll know who we can talk to about those issues. And I think a third thing I wanted to point out is that our program is going to be structured a little bit differently than usual tonight. We will carry you along with how that's going to occur as we go through the program, but hopefully you'll find it interesting and useful. One other thing to say before we start is that on your seats you will have found a yellow page, which is for your evaluation of the program. Those of you who are new especially, those are very important to us. They give us an idea of whether our programs have been useful and effective. As I said, tonight we're doing something different. We want to know if you liked it, if it was worthwhile and helpful, or if not. And so we can judge what we should do with our future programs. Our immediate plan tonight is that I'm going to be giving the opening presentation this evening, which will give you an introduction to the topic. The moderator this evening is Mary Devereaux, who many of you who've been to past programs know. Mary is a philosopher and an ethicist. She's been with the Research Ethics Program and involved with medical ethics at UCSD for on the order of about a decade, I guess would be about where we should place that. And she is going to help us work on this evening's topic, as I said, in a different way than usual. And rather than tell you too much about that now, I will just come back to that. We'll come back to that after my presentation. So let me switch to the presentation here. Whoops. And realizing that I had done that wrong, let me just switch this over here. So in addition to being co-director for the Center for Ethics in Science and Technology, I also have found my way into being the co-director for the Embryonic Stem Cell Research Oversight Committee at UC San Diego. And in that role, beginning at the earliest stages after California, through Proposition 71, decided to fund stem cell research to the level of $3 billion, I became increasingly involved not just locally but statewide in talking about some of the ethical and regulatory issues that go with stem cell research. As the research has developed, and developed at an incredibly rapid pace, and as the regulatory and the legal environment has also changed rapidly, we are continually looking at what are the questions that we should be thinking about. And one of the questions that is clearly on the mind of many people now is, what about clinical trials? If we're going to go from this human embryonic stem cell research, which we hoped would be the way to find cures or treatments for a variety of diseases, we clearly have to first try those things, those treatments in humans, those interventions. And if we're going to do that, that means a clinical trial. And so what I'm going to be talking about tonight in our program tonight, the focus is on what we should be thinking about as we go into those clinical trials. And the simplest way to look at this is that there are two possible avenues. One is to say, this is no different than anything else we've done for any other kind of clinical research. The other approach is to say, there is something different here for a variety of possible reasons. So in going through this, I'm going to begin by saying something about clinical trials that should be obvious to everyone, but since it isn't even obvious to the researchers who conduct the clinical trials, I want to start with that. And that's that the reason we conduct a clinical trial involving human subjects is because the treatment or intervention we are testing is not known to be safe or effective. We don't yet know if it will be safe. We don't yet know if it will work. And that's why we're doing this, which means that there is a risk if you are one of the first people in that trial. There's a risk you might be harmed, and if you were hoping to be cured or helped by the treatment, you may not be. And in fact, it may even be worse. There's a good chance that it will work because hopefully a lot of work has been done beforehand, but we don't know definitely. And under those circumstances, we have a couple of obligations. One is we need to figure out what standards we're going to follow before we decide to go into human subjects with that treatment. What do we need to know before we say that's good enough to try this on someone else? The second issue that we need to think about in such trials is to be sure that those subjects, when they enter the trial, know what they're in for. This process is often called informed consent. And what we're looking for is that somebody who's going to basically take those risks, that they know what risks they're taking, they know what they're in for, so that before they make that choice, they can decide, perhaps, I don't want to do this, before they get involved in your trial. So should we consider stem cell research as being different? Well, the biggest way that it's different, actually before I even go to that number one, is I should say stem cell research is different first because we've been talking about the human embryo. And that's a conversation that we won't spend a lot of time on tonight, but is something that is always in the back of our minds, and it's always really a reason why we have so much intense review and so much regulation around this subject. For some people, the use of the human embryo is tantamount to taking a human life, a child, and killing that child for the purpose of stem cell research. For other people, for the scientists, and for the majority of people in the public, it appears that they view that human embryo not as a human life, as a potential human life, but not as a human life. Instead, they view it as cells, human cells that provide us with an opportunity to do something for clinical research purposes. So on either side of this debate, we have people who would say it's morally wrong to use that human embryo because it's a human life. On the other side, people say it would be morally wrong to not use that human embryo because it's just cells that give us an opportunity to help better understand diseases and perhaps prevent or cure diseases. So that's always in the background is why stem cell research is different, at least this kind. But also, unlike many other interventions, not all, but many other clinical interventions, involve, for example, giving somebody a drug. You give them a pill. They take the pill on Monday, Wednesday, and Friday, and they're supposed to do that for 3 months. Well, if they decide after 1 month that they don't want to continue in the trial, they can say, I want to stop. And they can stop taking the pill, and they'll be done. But if you take some cells and stick them into someone's brain in order to see whether it helps prevent Alzheimer's disease, they can't say a month later, I've decided I don't want those cells in my brain. I will tell you right now that would be very hard to go in and remove those individual cells from their brain. There are scientists working on ways to inactivate those cells, but that is still something that is far off. So the fact is that probably for the foreseeable future, most of what we're talking about means if you're going to take a cell that came out of human embryonic stem cell research and put it into a human being, that's irreversible. So this means we might want to consider a different standard for this research than other kinds of research. One way to look at the questions I've just raised is to say that it's risky. You put those cells in somebody, so maybe we should just not do the research. Well, that's almost for no one is that clearly an acceptable avenue. We are challenged with numerous diseases and disorders that we would like to do better with, and the way to get there is through further research, and that research ultimately means clinical trials. So for most people, the option is that we should not stop the research. But we should follow some standards in the process of that medical research. One principle that is a typical principle, I'm sure many of you know, the physician's principle, that you should first do no harm. So we shouldn't intentionally be doing harm in the process of what we do. And secondly, we shouldn't avoid the chance to do something good solely because of the possibility of harm. The fact is, every clinical trial has a possibility of harm. Every time we want to try something new, there's a possibility it won't work, that it won't be safe. And if we are going to accept the premise that we should advance medical knowledge, medical understanding, then clearly we probably have to move forward. We just have to figure out how best to move forward. With stem cell research, the side effects that might occur, the harms that might occur, are not just a possibility, but they are to some degree inevitable. The goal of the intervention in almost all cases is to change body function. Otherwise, why would you be putting the cells into someone's body? And if the goal is to change body function, the likelihood that we'll be able to change just one thing that we want to change and nothing else is extraordinarily low. The body is very complex, and everything we might want to do might have multiple effects in the body. And second, it would be remarkable if there were no related effects in non-target dimensions. In other words, things that we weren't intending. So some of the things might occur because of what we intended, but some things might occur because of things we didn't intend. We don't intend, for example, one of the options now would be to take cells that have been created by the use of a virus that allowed us to modify those cells to be what they are. If we put those cells in the body, those cells might do what we want, but the virus might do something that we don't want. Or we might put the cells in the body, and in the process of doing that, we might simply cause damage to tissue around those cells as we try and insert them into the body. So there are a lot of things that are likely to happen that will be risks or harms with the things we try to do. Those unintended mechanisms of effects of what we're trying to do, things that we didn't anticipate, all mean that there's an increased risk of side effects. Some of the examples of this that one I just mentioned is tissue damage due to surgical insertion, but others include things like tumors. If we end up inserting human embryonic stem cells, which are by definition cells that will readily proliferate, divide, and become new cells, there is a high risk that tumors might form. And depending on how these cells are formed themselves, there's other reasons why we might get tumors forming. Because the cells proliferate, they divide too often. They might go into the wrong areas of the body and produce effects that we didn't intend. Those cells might change. They might backtrack. If we had a cell that was, for example, a nerve cell, maybe depending on how we've created it, it will have the ability to backtrack and become a cell that could become other kinds of cells. That means to de-differentiate. Or it might further differentiate. We might put a human embryonic stem cell into the body, and it might change into a liver cell, or a kidney cell, or a heart cell, or something else. And any of those cells may be producing effects that we didn't intend. This particular case is a good time to, as an aside, note many of you have heard, I'm sure, of treatments, often in other countries, where somebody says, we're going to give you these stem cells, these wonderful cells that can become any cell of the body. We're going to give you a treatment with these cells. We're going to stick them into your body and fix the problems, such as with your brain. You've got some sort of brain disorder or a kidney disorder or something else. But if you think about it for a moment, that is almost preposterous. I won't guarantee it's preposterous, but it almost is, that you're going to take this cell that can become any cell of the body and inject it into somebody's body, and it's going to decide somehow to only become repair the damaged cells and not do something else in the body. So we have all of this that becomes uncontrolled. I mentioned the viral vectors, the coincident introduction of those, and there may be immunological reactions to the cells you put in the body, which can be a worry if the cells aren't if the recipient is different immunologically than the donor, then there may be reasons why we get something different. So with all those reasons, what should we do? Those are some problems that we might have. It's not that these are new problems. Many of them have occurred with other kinds of treatments. But what should we be reasonably thinking about? That's our topic for tonight. The first one, and the things I'm going to go through now should not be taken as the gospel truth. They should not be taken as the final answer. They should be taken as some random meandering thoughts of Mike Kalichman on some things that you might want to consider. We're going to ask you what you think is important. You may think some of these are important. You may think there's some other things that I've missed. But these are some examples of the kinds of things we would like you to be looking for and thinking. So the first one is that we shouldn't do this work in humans until we have good evidence from animal studies that it's going to work. And for an intervention that is likely to be irreversible, I would go further and propose that we should raise the bar beyond what we normally do for other kinds of clinical trials. We shouldn't just say some animal studies. We should say those animal studies have to provide us with at least somewhat more evidence than we normally expect. So we're raising the bar so that we have more reason to believe that it will be safe and effective based on what we learned from the animals. Second, we would like to think that when people are preparing to do clinical trials with a new drug or a new treatment, that they have good reason to believe that there is a mechanism by which it will work and it will work well. The unfortunate news for you is that many things that we use and that we test, we don't actually have that good reason for an effective mechanism. We've learned that it is likely to work, and people say, well, we have reason to think it will work. It's important. As long as it's safe, we can go forward with it. Now, to some degree, there's more and more of a push when trials are done now that we do have to look for some mechanistic argument. But what I'm arguing here is that if you're looking at something that is, again, irreversible, we should have a standard that raises that bar higher. We should have good reason to believe that there's a mechanism by which what we're trying should reasonably work. We shouldn't just say, well, there's stem cells. Stem cells are wonderful, so we put them in the body, they'll help. Third, if we're going to start doing this, the question is, who should be the first people that are getting these treatments? If we're going to be trying stem cell therapy for the first time, where should we be trying it? One option would be to say, let's go to people who are not very sick. Let's say I've got a treatment for Alzheimer's disease. And if I want to do that, if I go to somebody who's sick, they might die more easily or have problems. They're already compromised in various ways. Maybe I should go to people who don't have Alzheimer's disease yet and start the treatment. And we'll do the treatment for 25 or 30 years and see if we protect them from Alzheimer's disease. Well, there are a number of flaws with that approach. One of them is that those people, because we're going to wait 25 or 30 years, may or may not develop Alzheimer's disease, but that means 25 or 30 years before we get an answer. And the second thing is that we're taking something that we don't yet know how safe or effective it is. We're trying it in a population that may never get Alzheimer's disease. And it's a little bit hard to rationalize that trade-off. The different approach that often is taken for many new treatments is to say we should go to the people who are the sickest. We should go to the diseases that are the most problematic. And especially at the beginning with this type of treatment where we don't yet know whether it's going to work, it would seem a bad choice to go to people who are otherwise healthy and take the risk that they'll start getting tumors and cancer or something else because of something we haven't worked out yet. So the argument here would be to restrict those initial trials to diseases and disorders with high levels of mortality or morbidity, and that could help us balance the unknown risks against what we already know are the known harms of the particular illness. So we're saying, given that they're already so bad off, we might be able to do this now and see what happens before we go on to others. Fourth, I'm proposing a robust informed consent process. This is one that I haven't seen from anyone else, or they haven't used this terminology. What I'm arguing for is something that goes beyond the informed consent for the potential research subject. I'm saying that we need informed consent for everybody in the process. The subjects, the patients that might get this treatment later, need to be involved in an informed consent process. The families of those patients and subjects, the physicians who are treating them, and even the researchers who are conducting the clinical trials, because my sense is that all of these groups are not getting it, that we don't know if this is safe and effective. And particularly since we're now looking at something that is so new, that's moving in a new direction, we don't even know what we should really be watching for. If I take some cells and put them in your brain, what should I be looking for as possible neurological, neuropsychological side effects? We could run a complex battery of tests on everybody, but we'll be testing for the things we think of first. We just don't know. So all of these people involved, if they knew it was uncharted territory, will be asked to be alert to something that doesn't look right. My partner, my husband, my spouse, my wife, whoever it is acting funny. And they don't usually act like that. So you go tell the doctor that way. Usually they're pretty serious. So the idea is to let people, is to make sure that we get that data, get that information, and that informed consent process would help us all. Fifth, by the way, there's only 6 of these in case you're wondering how long this kind of goes on. So, and actually I've got 37 of them. What I just described is going to be anecdotal information. Basically, this is not proof. If I go in and say, my wife was telling jokes when she didn't tell jokes before, I'm worried that this is a side effect of this treatment, then that's just an anecdote. It's one story. It's not a proof. What we want is we want to be collecting that anecdotal information so that we know where the problems might be, to collectively see what the what's going on. And then 6th, and the key here, and I'm going to then have a concluding statement after this, is that we should then, after that, do prospective studies to now design a new study to test where we've identified plausible risks. Based on the collected anecdotal information, when we see something that's coming up, that's when we move to the next stage to do something more. And so the decision to do that will be based on what we learned from the animal studies, what we worked out about the mechanisms, what we saw from the anecdotal observations. So we decide when to try and test to see what the issues are. So I'm calling this 7, but it's kind of a summary point that we should balance the risks. So one way to look at this is ignorance of the special risks of these interventions risks serious setback to the field of research. So if we just ignore those risks and say, you know, I want to solve the Parkinson's disease problem as soon as possible. Let's not worry about the possibility that something might happen wrong here, because the sooner we move ahead, the sooner we can find out what doesn't work, and the sooner we can find out what does work. And there's a lot of compelling truth to that argument that makes a lot of sense. But the worry here is that if we do plunge ahead too quickly, what we're going to do in that process is potentially have something that's really bad happen. And if that happens, it may set the whole field back. We may all say we've got to pull back from further clinical trials for a year or 2 years while we sort out something that had happened because we hadn't thought it through. So that's what we are balancing on one side. But on the other side, if we worry too much about these risks, if we place too many hurdles in front, then the research won't move forward either. So we have a delicate balance to be accomplished here. And that delicate balance can't be solved just by the scientists working in their laboratories and deciding what they think is the right next thing to do. This is something that needs to involve the public, and tonight will involve you. So we are left with this situation. Either of those errors risks slowing this promising new technology, restricting opportunities for clinical studies, and denying patients the opportunity for new therapeutic options. So we want to try and do as well as possible. We need to figure out how we should do that, how we should negotiate that process. So I'm going to summarize my recommendations that I just gave you, and we're going to be asking you in a moment. Mary Devereaux in a moment will come up and tell you how we're going to do this. We're going to be asking you in a moment to help us figure out what that should be. And this is not an idle exercise. Very few people have so far been asking, what should we be doing before we go into clinical trials? So as I've proposed that we should raise the standard of evidence in terms of animal studies and knowing the mechanisms, that we should conduct those first phase 1 trials to see if it's safe. We should be conducting those trials in people who have the worst disease, the most difficult situations. We should enhance the informed consent process so that it's not just for the subjects, but for everybody involved, so they are all alert to what is being tried and what risks we're facing. We should collect that anecdotal information and then develop evidence for the possible adverse events that we think may be there so that we can determine where the problems are and where they aren't. And finally, this whole process is necessary to balance the risks. So that's my summary, sort of introductory tonight.